they can have up to a dozen layers of fungicides, pesticides, nutrients, um, you name it, they have it. And each one of these colors here represents a different type of seed with a different type of treatment. So one of the things that they'll have in there is biopesticides or microbes that assist with nutrient uptake, so rhizobacterium or bacilli that will assist in that, nitrogen fixation. And again, it's a huge industry, and there's constantly searching for the next better microorganism to become part of these types of seed coating. There's waste control, such as thatch reducers, thatch being basically what's left over in the field. And sometimes in agriculture, once they're finished in the field, they either have to plant a new crop or they have to just move over a couple feet to the left and plant under each row because the material gets so thick in there, it's impossible to plant more. So there are bacterial inoculants that people use to help break those down quicker. There's actually a bacterial product out there that you probably ingested. It's been used for 20 years on apples. I'm not trying to sway you away. Obviously, they wouldn't be doing it if it wasn't something that wouldn't hurt you. But there's a Pseudomonas syringae that is used as a antifungal product that's sprayed on many apples and pears post-harvest. It's one of the very few that's used post-harvest. Most of these are used during the growth of the plant in order to prevent, the plant itself from being, hurt by pathogens. In this case, it's sprayed on the apples post-harvest to prevent any microorganisms from growing in bruises. So you can imagine, as that apple's tumbling down the line in the packing house, it's going to hit maybe here and there. A little bruise and fungi will get in and start just going to town on all the sugars in there and cause big brown spots in your apples. This microorganism will actually go into that small wound and through competitive exclusion grow in there and prevent the fungi from coming through, but not further cause a large bruised area. So just another application that may come as somewhat of a surprise. But again, all of these companies who are working with these products are going to need people familiar with microbiology, molecular biology, and so on. So that's kind of a roundabout of what some of the markets that are out there when we're talking about live microbes and the use of live microbes. The other thing I wanted to go through was just our company and kind of how we're set up. It's not too much different than many of the other companies out there. But I also wanted to open your mind to the fact that there are career paths not only in the lab, and I think most of us, especially in graduate school, think we're ultimately going to be in the lab scenario. And myself, I did, and then wind up becoming a manager of some sort, and at this stage in my career, vice president. So there's other areas that you'll see that, from a lab perspective, you may fit better in something other than that. And you may never know until you kind of fall into it. It's kind of the way life works. But so just an overview of what our company is comprised of. We have administration, sales, accounting, purchasing, production, sanitation, fermentation, regulation, QA, QC, and R&D. And there's several of these that would allow for someone with technical experience to thrive in and actually are required part of. So the first one is probably the most obvious, being quality assurance, quality control lab. We have one on site here. We have several technicians with microbiology and molecular biology backgrounds that will work in the lab. The techniques involved there, as I'm sure most of you are familiar, working in labs, media solution and preparation, microbial techniques, molecular biology techniques, the use of scientific instrumentation. And again, this is something that's going to change from lab to lab you go in. Chances are right now you're working on the project, your graduate research project, and you're kind of focused in on what you're doing. To me, it's always about not exactly what you know, but the fact that you learn new instrumentation, new processes. So always be thinking in that matter. When you're learning in college, in graduate school, it's always about problem solving. And that's from someone who would be looking to hire a new addition to my team. I would first look at their passion for what they would like to do, but also their ability to problem solve and work on new things. Are they excited about the opportunity to come in and work on new things, or are they simply, are they used to kind of being in this niche? Well, I can do this, and that's what I'm comfortable with. So, and that's good too in some cases. But just kind of be thinking about that as you're going around and looking for places to work. Another thing is just process comprehension. To me, it's more important to understand what you're doing than it is to remember it all. You can't remember everything. That's why we have Google. But to understand what you're doing to the very roots of the building blocks of what you're doing, I think is very important. In our lab, one of the key functions is strain maintenance. We have over 100 different strains of bacteria that will store cryogenically, some of which are ours, some of which are customer proprietary strains that we'll use only for their purposes. Interpretation of results is obviously important in a lab, and process validation in terms of QA. So in quality assurance, in any company, you have to make sure that what everyone else is doing is by the right way. QA is really the police of any given business. So not even the president can tell QA that they're doing something wrong. It would be quite the opposite. But it requires people who are technically skilled in these types of areas to understand it when you're in a technical business such as this. Just a couple slides on some quality control parameters that we have here. We do all efficacy testing on our products, so not only does it have to meet specification in terms of bacterial concentration, we have to determine its efficacy for that specific application or use. In the upper right-hand picture, we have a pseudomonad that will actually grow using the volatilized vapors from naphthalene crystals as its source. And naphthalene, if you've ever smelled mothballs or those old urinal cakes that they have, that's one of the volatile organic compounds in there. It'll volatilize on the top of that plate, and actually the pseudomonads will grow in minimal media with no other carbon source just by utilizing the naphthalene as a food source. The plate below there demonstrates what's a spirit blue agar. Basically, it's emulsification of a triglyceride. And if you have a bacteria capable of breaking down the triglyceride or at least separating it, you'll get a clearing zone. So again, I'm trying to mix this a little bit of what we do and what's out there, just to give you an idea of what it would be. R&D, again, another obvious one. New product development, strain selection and characterization, fermentation scale-up. Our third biggest area, and I'll go into it in a minute, is fermentation and contract fermentation. So we have companies who will come to us that they've been working on strains for so many years for their specific application, but they don't have a clue how to commercialize it, or even if it's commercially viable. They will present us with these strains, and it's best to do it in tandem with their trials, their field trials, their efficacy testing. And we have the capability here and the experience to take those strains, put them through various media formulations and process parameters for fermentation to determine if, and then if so, how to produce these cultures on a commercially viable level. Because no matter how efficacious a product is in its respective application, if you can't get the price to where someone would actually pay for it, there's no sense in moving forward on it. So there's a great deal of work involved in that there. The other thing with the microbial product is that you have to ensure that it's stable. So it's one thing to make it and put it in a package, but will that product retain its viability and its activity from the time that it's made to the time that it gets on it's being used in application. And again, some of this I kind of had for a previous presentation, and so I'm just scanning through it. So the other thing R&D would do is be creating protocols. So you not only develop what it is, the rest, say, fermentation. When you see our fermentation department, we have a department that's their sole job is fermentation. Now, they don't come up with the protocols. That would be a function of R&D and process development. So once we determine the best way to do it in the lab, you have to create the protocols so that someone can follow it very easily, and what I call making it foolproof, so that it would be almost impossible for someone to mess it up once we get on the larger scale. When you're talking things that cost hundreds of thousands of dollars, and in pharmaceutical industries, millions of dollars, you need to be sure everything is done exactly the way it's supposed to be done. This is just a slide showing some of the things that we're working on or have worked on with plant inoculants, soil inoculants, and some a type of bacterial product to break down, in this case, a fatty acid. And the third area, basically, like I mentioned, is contract fermentation. We do strain transfers here of cultures. Again, media process optimization, scale up. Another thing in fermentation is that you have to separate the culture from the fermentation broth. In some cases, the active compound could be in the fermentation broth, so depending on what you're after, you'll need varying levels of separation and purification. Again, they will have to then figure out how to stabilize it. We do freezing. We also do lyophilization with some cultures, like if you're producing bacillus spores, which are highly used in this industry. They can tolerate higher temperatures and pressures so that you can actually spray dry them into a powder. So there's a number of different ways to do that. But again, there's technical people required, people with scientific backgrounds required to not only develop all these processes, but see them through. I won't go into that. I'm going to get a little too much detail. This is just an outline of how one of our processes works for receiving strains. This is a process flow chart for how we handle cultures and how they go through our fermentation. So the lab, our QC lab, stores the cultures and maintains the cultures. We'll go through several stages of inoculations before we get to the fermenter. The fermenter then is grown up at a set time, set temperature, where we control oxygen agitation, addition of any other chemicals, whether it be sugars, acid bases for pH control, antifoam for antifoam control. There's a number of different parameters that need to be measured. We can measure those online with systems through PCs or in some cases our cell phones to ensure that the process is working thoroughly throughout the time it's supposed to be. At that point, we'll either process via centrifugation or filtration to remove the cells. The cells then go through a cryoprotectant mixing, and they'll go into a lyophilization, drying, or freezing stabilization. At that point, they go through a quality control hold too. So again, very important that nothing makes it out here, out of our these walls that has not been through the QC check. We need to make sure that our products contain the right levels of microbes, they're doing what they're supposed to be doing, and they're absent of unwanted microorganisms and pathogens. Another area that requires people with technical backgrounds is going to be regulation, and it's something that you'll very soon become familiar with once you go, if you're looking to go into industry. Every industry has its own degree of regulation, and it's very time consuming. But in order to do it and to be able to work through it, there has to be someone involved with technical background, so scientific type background, especially when it relates, particularly when it relates to a scientific type of process. So whether it's in pharmaceutical or applications such as the ones I've discussed today, they all have their regulation just to varying degrees. There's a couple listed there on the slide in front of you that I deal with. And again, just another career path that you may not have really thought of that could be available out there. And the last area, which I would have cringed at one day, would be technical sales. But I mean, there are certainly areas in which you need technical people to translate information. I always say you can teach a scientist to sell, but you can't teach a salesperson science. Not everyone's for sales, but there is a certain group of people that fall into that, and it is required. And to be quite honest, technical salespeople usually end up making a lot more money than the people at the bench. So if you're that type of person, I'd encourage you to look into that sort of thing. So that pretty much wraps up my presentation. I'm sure I've gone over whatever a lot of time I have, and I appreciate your patience and staying with me. I'm available for any questions. If you think of one afterwards, feel free to contact Dr. Teeter, or if he has any